Welcome back to BiochemSerye Learning on the Go.
Yeah.
This is our 9th deep dive.
Ninth one.
And today we are tackling a cornerstone of biochemistry,
Yes.
the 4 levels of protein structure.
It's,
Think of it as like a
big one.
deep dive into a protein's blueprint,
Yeah.
the guide that dictates how these amazing molecules are built
Right.
and how they function.
It's a topic we revisit every year.
Oh, for sure.
With our students,
Yeah.
both at the college and medical school level.
Right.
And trust me,
Yeah.
it never gets old.
Right.
Never.
It's like unpacking this complex puzzle where each level reveals a new layer of intricacy and elegance.
Absolutely.
But why is this so important for our listeners?
Well,
Especially those hitting the books for their biochem courses.
imagine trying to understand how a car engine works.
Right.
Just by looking at the whole thing,
Yeah.
you wouldn't get very far, would you? You
Yeah.
would need to break it down into its individual components,
Yeah.
see how they fit together, and understand their specific roles.
Right. It's the same with proteins.
Exactly.
We can't just look at the final folded structure.
Yep.
We need to understand how it gets there.
Yep.
Step by step,
Level by level.
level by level.
Exactly.
And that's why mastering these 4 levels is so crucial.
Right.
It's not just about memorizing terms.
Yeah.
It's about gaining a deeper understanding of how proteins behave,
Yeah.
how they interact with other molecules,
Yep.
and ultimately how they carry out their diverse functions in living organisms.
For sure.
Okay, so let's start with the foundation, the most basic level of protein structure, the primary structure.
So think of the primary structure as the protein's word. It's
Yeah.
a linear sequence of amino acids.
Yep.
Just like the order of letters in a word matters.
Uh-huh.
You know, cat is very different from act.
Right.
The precise order of those amino acids is absolutely critical.
So if we swap even just 2 amino acids in a protein sequence,
Yeah,
we could end up with a completely different protein.
absolutely.
Wow.
Let's take a simple example. Okay.
Alanine, arginine, glycine versus glycine, arginine, alanine.
Okay.
Same 3 amino acids,
Right.
but different order, different properties,
Mm-hmm.
potentially different function.
Interesting.
Yeah.
And this order, this precise sequence,
Yes.
is determined by what?
Our DNA, of course.
Okay.
The genetic code within our DNA
Yeah.
dictates the order in which those amino acids are linked together during protein synthesis.
So it's like having a blueprint that tells the cellular machinery.
Exactly.
Okay, first grab an alanine, then an arginine, then a glycine, and link them up.
Precisely.
Whoa.
And those amino acids are linked together by strong covalent bonds
Yeah.
called peptide bonds,
Okay.
forming the protein's backbone.
So we have this linear chain of amino acids held together by those strong peptide bonds.
Right.
That's the essence of primary structure.
Yeah.
But proteins aren't just floppy chains, are they?
No way.
Oh,
They
crap.
fold into these intricate 3-dimensional structures.
Right.
And that brings us to the next level.
Secondary structure.
Secondary structure.
This is where we start to see some local folding patterns emerge.
Local folding.
Yeah.
What do you mean by that?
Think of it this way. The primary structure is like a long piece of string.
Okay, a string.
Secondary structure is where that string starts to coil or fold in on itself in certain predictable ways.
So it's not the final 3D structure,
Right.
but it's starting to take on some shape.
Exactly.
Yeah.
And the 2 most common secondary structures
Yeah.
are alpha-helices and beta sheets.
Okay.
Imagine an alpha helix as a spiral staircase, a tightly wound coil,
Uh-huh.
and a beta sheet is more like a pleated sheet or a folded paper fan.
Okay. I can picture those.
Good.
So what drives the formation of these shapes?
Yeah.
Is it something about the amino acids themselves?
It's actually the hydrogen bonds,
Okay.
but not between the side chains of the amino acids.
Right.
These hydrogen bonds form between the backbone atoms,
Okay.
the ones that make up the polypeptide chain itself.
So those slightly positive hydrogen atoms on one part of the backbone
Yeah.
are attracted to the slightly negative oxygen atoms on another part, pulling the chain into those specific shapes.
You got it.
Okay, cool.
It's like an intricate dance of attractions and repulsions, all governed by those weak but numerous hydrogen bonds.
Right.
And these secondary structures aren't just for show.
Right.
They play a crucial role in stabilizing the protein and
Yeah.
setting the stage for the next level of folding.
Now, I remember from my own biochemistry days that there's one amino acid that's kind of a troublemaker when it comes to alpha-helices.
Yes, proline.
Proline. Yeah.
Its rigid cyclic structure throws a wrench
Yeah.
into those neat hydrogen bonding patterns
Right.
needed for a smooth alpha helix.
Yeah, it's like trying to bend a stiff piece of wire into a smooth spiral. It
Exactly.
just doesn't quite work.
Nope.
So proline acts as a helix breaker,
Right.
disrupting that smooth coiling.
Precisely.
Okay.
And those disruptions can actually be quite important. They introduce bends or kinks into the protein structure,
Right.
which can be crucial for its flexibility and function.
Okay.
Think about it. If a protein needs to be able to bend or flex
Right.
to interact with another molecule,
Yeah.
those proline-induced kinks
Yeah.
can provide just the right amount of give.
It's amazing how these subtle structural details can have such a big impact on a protein's behavior.
Absolutely.
Wow.
And that's what makes understanding these levels so fascinating.
Yeah.
It's all about connecting the dots between structure and function.
Okay, so we've got the primary structure, the linear sequence of amino acids. Then we have the secondary structure, those local folding patterns like alpha-helices and beta sheets.
Yep.
What happens next?
Right.
How do we go from those local folds to the final three-dimensional structure of a protein?
That's where tertiary structure comes in.
Tertiary structure.
The overall three-dimensional shape of a single polypeptide chain. Think of it like taking those alpha helices and beta sheets and crumpling them up into a complex, unique three-dimensional structure.
And what forces are at play in this intricate origami folding process?
Right.
Is it still just hydrogen bonds?
Hydrogen bonds are definitely involved,
Okay.
but it's a much more complex interplay of forces.
Okay.
We've got hydrophobic interactions where those nonpolar amino acids, the ones that hate water, trying to bury themselves in the core of the protein away from the surrounding aqueous environment.
So it's like they're trying to huddle together for warmth
Yeah.
away from the cold, watery world outside.
Exactly.
Okay.
And then we have hydrogen bonds, ionic bonds, and van der Waals forces,
Right.
all interacting between the side chains of those amino acids.
Okay.
They fine-tune the shape, attract certain parts of the protein together, and contribute to its overall stability.
And let's not forget about those disulfide bonds,
Right.
those strong covalent bonds between cysteine amino acids.
Disulfide bonds are like those heavy-duty staples that hold specific parts of the protein structure together.
Yeah.
They're especially important for proteins that need extra stability, like those that function in harsh environments.
Okay. This is all starting to come together.
Yeah.
So we have those local folds from secondary structure interacting and packing together, driven by those hydrophobic forces and various types of bonds to create this complex three-dimensional structure.
The tertiary structure.
You got it.
Okay.
And within that three-dimensional structure, we often see distinct regions called domains.
Domains.
These are sections of the polypeptide chain that fold independently
Okay.
and often have specific functions.
Interesting.
Like the protein has different modules that contribute to its overall job.
So one domain might be responsible for binding to a specific molecule, while another domain might be involved in catalyzing a chemical reaction.
Exactly.
Okay.
And these domains can be incredibly diverse in terms of their structure and function.
Right.
They're a testament to the incredible modularity of proteins. Wow. And how evolution has mixed and matched these building blocks to create proteins with a mind-boggling range of capabilities.
So we've covered primary, secondary, and tertiary structure. Is that the whole story? Is that all there is to protein structure?
For some proteins, yes.
Okay.
But for others, the story continues.
Okay.
They team up with other polypeptide chains,
Okay.
forming larger multi-subunit complexes.
Wow.
And that's where quaternary structure comes in.
So it's like a protein party.
I like that analogy.
Multiple chains coming together to create a bigger, more complex functional unit.
A protein party indeed.
Okay.
And just like at any good party, the interactions between the guests are what make it fun and interesting.
So it's the interactions between those individual polypeptide chains, now called subunits, that define the quaternary structure.
Precisely.
Yeah.
And just like with tertiary structure, those interactions are driven by a combination of forces.
Hydrogen bonds, hydrophobic interactions, ionic bonds, van der Waals forces, and those trusty disulfide bonds.
So it's like those same forces are playing a matchmaking game, bringing those subunits together in just the right way to create the final functional protein complex.
Exactly.
Wow.
And this teamwork, this quaternary structure, often leads to some pretty amazing functional advantages.
Like what?
Well, for starters, it can increase the stability of the protein complex.
Okay.
Multiple chains working together are often stronger than a single chain going it alone.
That makes sense. What else?
Quaternary structure can also allow proteins to bind to larger molecules.
Okay.
Or create those intricate binding sites that are just the right shape for a specific target.
Right.
And it can also lead to something called cooperativity.
Cooperativity.
Yeah.
That sounds intriguing.
Yeah.
What is that?
Cooperativity is when the binding of a molecule to one subunit actually influences the binding affinity of the other subunits.
Okay.
Imagine a group of friends sharing a giant pizza. If one friend grabs a slice, it might make the others more eager to grab a slice too.
I like that analogy.
Good.
So the binding of one molecule can make it easier for other molecules to bind.
That's the idea.
Okay.
It's like a chain reaction of binding events.
Right.
And it can make a protein much more efficient at its job.
So we've built our protein from the linear amino acid chain all the way up to the complex multi-subunit structure.
Right.
But proteins aren't just static sculptures, are they?
Right.
They interact with other molecules to carry out their diverse functions.
And that brings us to the fascinating world of ligand binding.
Ligand binding.
You can think of proteins as having these intricate pockets or surfaces, we call them binding sites, that are perfectly shaped to interact with specific molecules. Yeah. These molecules are called ligands.
Ligand.
So these handshakes or interactions are based on a combination of shape charge and those weak but numerous forces we've been talking about.
Yeah.
Hydrogen bonds, van der Waals forces, and so on.
It's like a locking key where the ligand is the key and the protein's binding site is the lock.
That's a good analogy.
Yeah.
But it's even more dynamic than that.
Okay.
Imagine a hand fitting into a glove.
Right.
The glove molds itself around the hand, and the hand can adjust its grip slightly to get a better fit.
Right.
That's more like what happens with proteins and ligands.
Okay.
There's a certain degree of flexibility and adaptability involved.
So how do we measure the strength of these interactions, these protein-ligand handshakes?
That's where we introduce two important concepts.
Okay.
The association constant Ka and the dissociation constant Kd.
Okay, I remember seeing those terms in my biochemistry textbook.
Yeah.
But I'll admit they always felt a bit abstract.
Sure.
You break them down for us.
The association constant Ka tells us how much the protein and ligand like to bind to each other.
Okay.
A high Ka means strong attraction. A tight bond, like those two molecules are really holding on tight.
So a high Chi means the protein and ligand are likely to spend a lot of time together.
Exactly.
While a low Chi means they're more likely to go their separate ways.
Exactly.
Okay.
And the dissociation constant Kd is the flip side of that coin.
Right.
It tells us how easily the protein and ligand come apart.
Mm-hmm.
A low Kd means they tend to stick together, while a high Kd means they're more likely to dissociate, to break apart.
So to summarize, high Chi, low Kd, strong bond, they like each other.
Exactly.
Low Chi, high Kd,
right.
Weak bond, they're not that into each other.
Perfect. You got it.
Okay.
And these constants are incredibly important for understanding how proteins function.
Right.
They tell us how strongly a protein might bind to a drug, for example.
Okay.
Or how efficiently an enzyme might bind to its substrate.
Now, I remember struggling with the difference between myoglobin and hemoglobin back in my student days.
Yeah.
They both bind oxygen, right?
Right.
But they have different roles in the body.
Yeah.
Can you help us clear up the confusion?
Absolutely.
Okay.
Myoglobin and hemoglobin are classic examples of how seemingly small structural differences can lead to big functional changes.
Right.
Let's start with myoglobin.
Okay.
It's a relatively small protein found in muscle tissue.
Uh-huh.
And its job is to store oxygen.
So it's like the muscle's personal oxygen tank.
Exactly.
All right.
And it's really good at its job.
Yeah.
Myoglobin has a single binding site for oxygen.
Right.
And it binds to oxygen very tightly with a very high affinity.
So it's like myoglobin is holding on to that oxygen with a death grip, not letting go easily.
Exactly.
Why is that important?
Well, think about what happens when you're exercising vigorously.
Yeah.
Your muscles need a lot of oxygen to keep going.
Right.
But the blood supply might not be able to deliver oxygen fast enough to meet that demand.
Right.
That's where myoglobin comes in.
Right.
It releases its stored oxygen when the oxygen concentration in the muscle tissue gets really low,
Right.
providing that extra boost of oxygen when it's most needed.
So myoglobin is like the emergency backup system, ensuring that muscles have access to oxygen, even when they're working really hard.
Precisely.
Wow.
Now let's contrast that with hemoglobin.
Okay.
Hemoglobin is found in red blood cells, and its job is to transport oxygen from the lungs to the tissues throughout the body.
So hemoglobin is like the delivery truck carrying those precious oxygen molecules to where they need to go.
Exactly.
Yeah.
And hemoglobin is a much larger protein than myoglobin.
Right.
It's actually made up of 4 subunits, each with its own oxygen binding site.
So hemoglobin has 4 hands to grab onto those oxygen molecules.
That's a good way to think about it.
Yeah.
And here's where things get really interesting.
Okay.
Hemoglobin exhibits cooperativity.
Cooperativity.
Yes.
Like those friends sharing the pizza.
Right.
The binding of one oxygen molecule makes it easier for other oxygen molecules to bind.
Precisely.
Yeah.
When one subunit of hemoglobin binds to oxygen, it actually changes the shape of the entire protein complex in a subtle way, making it easier for the other subunits to bind to oxygen as well.
So it's like a chain reaction of binding events.
Exactly.
Okay.
And this cooperativity is crucial for hemoglobin's function.
Right.
It allows hemoglobin to efficiently load up on oxygen in the lungs,
Yeah.
where the oxygen concentration is high,
Right.
and then release that oxygen in the tissues where the oxygen concentration is lower.
I'm starting to see how this all fits together.
Good.
Myoglobin, with its single binding site and high affinity for oxygen, is perfect for storing oxygen in muscles.
Right.
Hemoglobin, with its 4 subunits and cooperative binding, is ideal for transporting oxygen throughout the body.
Exactly.
It's amazing how these structural differences translate into such distinct functional roles.
It is.
Wow.
And it highlights the incredible versatility and adaptability of proteins. They're like nature's molecular machines, capable of carrying out an astonishing array of tasks.
Right.
All thanks to those 4 levels of structure we've been discussing.
You mentioned earlier that there's even more to protein structure than those 4 levels.
Right.
You said something about folds.
Remember how we talked about domains being those independently folded units within a protein's tertiary structure?
Yes.
Well, sometimes those domains arrange themselves into larger, more complex structural patterns called folds.
So it's like an even higher level of organization beyond those 4 basic levels.
Exactly.
Wow.
And these folds aren't random. They're
Okay.
often associated with specific functions.
So?
And they can be found in a wide variety of proteins, even those that have very different overall structures and functions.
So it's like nature has a toolbox of folds that it uses to build proteins with different capabilities.
That's a great analogy.
Yeah.
A toolbox of folds and domains that evolution has mixed and matched over billions of years to create the incredible diversity of proteins we see today.
Do you have a favorite example of a protein fold?
Well, one that always fascinates me is the actin fold.
Yeah, okay.
It's named after actin, the protein that forms those long filaments that make up our cellular skeletons, those internal scaffolding that gives cells their shape and allows them to move.
So the actin fold is found in actin, but it's also found in other proteins.
That's right.
Interesting.
The actin fold also shows up in proteins like heat shock protein 70, or HSP70 for short,
Okay.
which acts as a kind of molecular chaperone helping other proteins fold correctly.
Oh.
And hexokinase, an enzyme that plays a key role in breaking down glucose for energy.
That's amazing.
Yeah.
So even though those proteins have very different jobs, they share a common structural motif, this actin fold.
Exactly.
Interesting.
And it highlights how evolution can repurpose successful structural designs for different purposes.
Right.
It's like taking the basic design of a wheel and using it to build everything from cars to bicycles to skateboards.
This is blowing my mind.
Yeah.
It's like realizing that there's a hidden order, a deep structural logic underlying the incredible diversity of the protein world.
Precisely.
And that's what makes studying protein structure so rewarding.
Yeah.
It's like peeling back the layers of complexity to reveal the underlying elegance and beauty of these molecular machines.
I think it's time for a little review session to make sure our listeners have absorbed all this amazing information.
Sure.
You ready to put on your professor hats and quiz us?
Absolutely. Let's see if you've been paying attention.
All right, Professor, hit me with your first question.
Okay, let's start with a fundamental one. What type of bond primarily stabilizes a protein's primary structure?
Primary structure, that's the linear sequence of amino acids. So the bonds holding those amino acids together would be peptide bonds.
Correct. Peptide bonds are those strong covalent bonds that link amino acids together, forming the backbone of the polypeptide chain. They're the reason why the primary structure is so stable, providing that strong foundation for the higher levels of structure.
Okay, I'm ready for question number 2. Bring it on.
All right, how about this? What's the main difference between alpha-helices and beta sheets?
Ooh, that takes me back to my early days in the biochemistry lab, trying to visualize those structures. They're both secondary structures stabilized by hydrogen bonds. But the difference lies in the patterns of those hydrogen bonds, and the overall shapes they create. Alpha helices are those tightly coiled structures like a spiral staircase with hydrogen bonds within the same strand. Beta sheets, on the other hand, are more extended sheet-like structures with hydrogen bonds between adjacent strands, like a folded paper fan.
Excellent. You've captured the essence of those key differences. The hydrogen bonding patterns are crucial for creating those distinct shapes, and those shapes in turn influence how the protein ultimately folds into its tertiary structure.
All right, what's next on the exam?
Let's test your understanding of the hydrophobic effect. How does it contribute to protein folding?
The hydrophobic effect. That's all about those non-polar amino acids, the ones that hate water. They're like those kids at a party who cluster together in the corner away from the crowds. In the case of proteins, those hydrophobic amino acids try to bury themselves in the core of the protein away from the surrounding aqueous environment. And this clustering drives protein folding, creating that compact hydrophobic core, while the polar water-loving amino acids are happy to stay on the surface interacting with the water molecules.
Perfect explanation. It's all about minimizing those unfavorable interactions between the hydrophobic amino acids and water.
Okay, 3 down. What other protein structure challenges do you have for me?
How about this? Give me an example of a protein with quaternary structure.
Oh, I've got a classic example for you. Hemoglobin. With its 4 subunits, 2 alpha chains, and 2 beta chains, all working together to bind and transport oxygen throughout the body.
Spot on. Hemoglobin is the textbook example of quaternary structure and how those subunit interactions can lead to enhanced functionality like cooperative binding.
Okay, I'm feeling pretty confident so far. What's the next question?
All right, let's see if you can differentiate between a structural domain and a functional domain in a protein.
This one requires a bit more nuance. Let me see. A structural domain is a physically distinct region of the polypeptide chain, kind of like a self-contained unit. A functional domain, on the other hand, is a region that's responsible for a specific activity, even if it's not physically separate from other parts of the protein.
Excellent. You've grasped the key distinction between those two types of domains.
Okay. Ready for the next challenge?
All right, here's one that often trips up students. Why are disulfide bonds important in protein structure?
Disulfide bonds. Those are those strong covalent bonds that form between 2 cysteine amino acids. They're like those little molecular staples that hold specific parts of the protein structure together. They provide extra stability, especially for proteins that need to function in harsh environments.
Exactly. They're like those reinforcement beams that keep a bridge from collapsing.
Right, give me another one. I'm on a roll.
All right, how about this? If a ligand has a high association constant, Ca, for a protein, what does that tell you about their interaction?
A high association constant, or Ca. That means the ligand binds tightly to the protein. They have a strong affinity for each other, like those best friends who are always hanging out together.
You nailed it. A high Ca means they're likely to spend a lot of time together forming a stable complex.
Okay, what else do you have for me?
All right, here's one that should be familiar. Why does proline disrupt alpha helices?
Proline the helix breaker. It's all about its unique structure. Proline has that rigid cyclic structure which makes it unable to adopt the proper bond angles needed to fit into a smooth alpha helix. It's like trying to force a square peg into a round hole. It just doesn't work.
You're absolutely right. Proline's rigidity throws a wrench into the helix party, often introducing bends or kinks into the protein structure.
Okay, 2 more to go. What's next?
Can you give me an example of a protein fold?
Let's see. Oh, I remember. We talked about the actin fold earlier. It's found in actin, of course, but also in other proteins like HSP70 and hexokinase, even though those proteins have very different functions.
Excellent. The actin fold is a classic example of how nature can repurpose a successful structural design for a variety of purposes.
Okay, final question. Bring it on.
All right, here it is. How does the quaternary structure of hemoglobin contribute to its function?
Hemoglobin's quaternary structure, those 4 subunits working together, is what allows for cooperative binding of oxygen. When one subunit binds to oxygen, it changes the shape of the entire complex, making it easier for the other subunits to bind to oxygen as well. This cooperativity is essential for hemoglobin's role in transporting oxygen from the lungs to the tissues.
Fantastic. You've aced my protein structure exam.
I told you I was ready for this. It's amazing how all those levels of protein structure work together to create such a diverse and functional set of molecules.
It really is, and this is just the tip of the iceberg. There's so much more to explore in the world of protein structure and function.
Well, I think we've given our listeners a great foundation for understanding this essential topic. And for those who want to delve deeper, we'll include some additional resources in the show notes.
Great idea.
And on that note, we'll wrap up this episode of BiochemSerye Learning on the Go. Until next time, keep those brains buzzing and don't be afraid to explore the fascinating world of biochemistry.